So, hello, I'm starting my PhD in the small biosystems lab here in the physics faculty in the UB. And during my PhD, I will be concerned about evolution. And I will try to quantify an evolutionary process in the lab. So first of all, what is evolution, or what are the basic features of evolution? So one has to bear in mind that when we speak about biological evolution, we're not speaking about one single element, but about a bunch of elements, a population. And within that population, the elements show some variation. That is, there are small differences in the elements that can be selected. And because of selection, the variants that are better at performing some task will be enriched in the next generation. And then that will give rise to a directional process that will end up in a structure that is good at the function it has been selected for. So that takes us to another important concept in biological evolution, that is heredity. And that means that the elements within an evolving population must be able to transmit their features to the next generation. That is, they must be able to copy themselves. And then the elements in a biological evolving population must have a genotype. That would be the information, the basic information necessary to produce a phenotype. So here in this drawing, the genotype would be represented by the sketch of the crocodile, the basic information, and then this information unfolds in space and time to produce the real crocodile that can move and do things. So this is what I will try to reproduce in the lab. And to do so, I will use RNA molecules. Why? RNA molecules can replicate, so the heredity condition is fulfilled. And then RNA molecules can also mutate. So I will create a bunch of mutants, and instead of having an homogeneous population of molecules in the tube, I will have this other situation where molecules can be of different types. Then also RNA molecules can fold in a three-dimensional structure, and the different genotypes will give rise to different structures. So instead of having a bunch of linear molecules, I will have a population of different structures. So as we saw here, RNA molecules are interesting because they have this genotype and phenotype in the same molecule. And then because of that phenotype, because of that structure, RNA molecules can perform a task, and I represented it here by binding to that blue structure. And you can see here that some of them are better than others to binding to that structure. So blue molecules are better than yellow molecules. So instead of having, at the end of the process, instead of having this initial concentration where all molecules are equiprobable, I will have a population enriched in those variants that are better at binding the blue structure. So this situation here. And I said I would quantify an evolutionary process. So first of all, I will try to characterize the phenotype distribution, and I will do so using optical tweezers. So optical tweezers will be interesting in this project because they allow you to characterize RNA molecules one by one. And that will give a precise characterization of the structure of the molecules in the population. So I will attach my RNA molecule to two beads. I will immobilize one in a pipette, as you see in the figure, and the other one will be trapped in a laser. And by moving the laser, I can perform some force on the molecule, and that will give me information about the structure of that molecule. Then, by sequencing, I will characterize the genotype distribution of the population, and I will know which amount of information is present in that population. And because we are speaking about population and there is variation within that population, it makes sense to quantify this variation, and the physical quantity that quantifies variation is entropy. So I will calculate entropy and see how it evolves in time. So to finish, I would like to leave you with an image I found while preparing this presentation. So evolution is indeed a theory, and we have the theoretical and experimental tools to explore it. Thank you.